Okay, we're coming down the home stretch. I'm glad that everyone has stayed. Thank you, as Doug says, he makes up for going in early by staying late. So thanks everyone for staying. I also realized that we made a mistake that we didn't put a microphone in the middle of the room so that Doug could clear his throat and give us insights into our work that we had never thought about before. We have all been there, right? And as you could tell today by the breadth and the depth of all the talks, the history of HIV research is basically, you could read it through Doug's CV and then through the work of his trainees, and I really appreciate that. And I didn't know any of that when I came to talk to Doug as a fellow in 1999 or 2000, at the end of 99, 2000, trying to figure out what lab or what work I wanted to do. And I said, hey, I'm an old country doctor, as Doug said, and had no idea what in the world a lab was and what I was going to do. And he's like, oh, we'll teach you which end of the pipette to put in your ear. And he did. And I'm going to talk a little bit about other things and techniques that he taught me. So, as if the way that one died mattered is what we're going to talk about today. And the quote that I like for this talk is, "My, you chivalric fool." So these two guys in prison, they're getting ready to be beheaded. Says, "As if the way one fell down mattered." And the other one says, "Well, when the fall is all there is, it matters." So these are some questions that I want you to think about as we go through this talk. And these are actually questions that we gave to a few people, so we'll talk about that later. But would you consider participating in a research study that would potentially help others but not yourself if you thought you had less than 6 months to live because of a terminal illness? So would you consider participating in a research study that would potentially help others but not yourself if you thought you had less than 6 months to live because of a terminal illness? Would you participate if it might decrease your lifespan further, and if so, how long? What physical discomforts would you ever consider enduring for participation? Nausea, vomiting, headache, blood draws, listening to me talk. Would you participate if it exposed you to an infectious agent that might cause you to become infected with that agent? Would you participate if it exposed you to an infectious agent that might cause you to become infected with that agent? So we interviewed a bunch of people as well who had HIV with a terminal illness, and we were preparing for this study called The Last Gift. And we asked them, would they be interested in participating in the study at the end of their life? And a lot of very interesting quotes is what we got back. "I feel like the last few weeks are wasted. I hate my HIV, but I hate my cancer more. I wish I could do more. I used to participate in research all the time, HIV research. I would like to leave one last gift to my community." So we're going to put that on the side for just a second, and we're going to talk a little bit about HIV reservoirs in the tissues. As you've heard a lot about this afternoon, how HIV persists and remains active or remains silent within the body, only to return when therapy is stopped, is a very important goal to understanding if we're ever going to cure HIV. It's established during primary HIV infection, so within the first week or two that someone's infected, there's a very solid reservoir that's already there. It might be really small, like Marta talked about, but it's still there. And it's in anatomic sites, like Joe talked about with gut, GU tract, everywhere you look. And it plays this sort of hide and seek with the immune system and antiretroviral therapy. But when the therapy is stopped, the virus comes back. And we wanted to understand the size and distribution and the dynamics of these reservoirs. But the problem is that studies and living people looking at all these deep places where HIV hides is not easy. But there are some autopsy studies. It's been around since the very beginning of HIV research. But there's usually not as good collection and characterization of data before someone passes away. And then the autopsies are rarely performed quick enough to really preserve all the molecular protein information that we want to collect. So we thought about this study called Last Gift, and we were interested in recruiting people who are altruistic, who had HIV and a terminal illness. So they might be going to die of cancer or cardiovascular disease or ALS. And we enrolled them, and we followed them closely to collect clinical data and regular sampling, blood, genital secretions, stool, and then we perform a very rapid autopsy to collect tissues, all within about 6 hours of death. And we really want to preserve as much information and cellular information that there is there. So we started in 2017 from people who had HIV, a terminal illness, and they were going to die of a non-HIV related condition. We took detailed reports of their social clinical factors. We collected all their previously collected biological samples. San Diego, thanks to Doug and others, has a wide breadth of HIV research that's been going a long time, and we have lots of freezers with lots of specimens and hunting down those specimens that might have been collected even decades earlier. And then we collect their full bodies when they pass away. Here's our team, that many of the people in the room maybe or may not can recognize them at one of our autopsies. So I'm there in the middle somewhere. Which one is this button? There we go. So we go through, there's a body on this side, and there's a diener who takes different pieces and gives us like, here's the spleen, here's the ileum, here's the prostate, and then it's brought over here in little ice trays, and then the cutters, including me, take little pieces of it, and we put it in little tubes and that are already labeled by Laura, and so that we can store them appropriately and flash freeze them, etc. So we have lots of different ways to collect these specimens, very valuable specimens that we can use later. Oh, sorry. So here's our cohort. I think we have more people now, but we have, at this time, it was 11 individuals. Here's all the different diagnoses that we have. Some people at the end of their life want to stop their therapy, so we have some people who stop and some people who don't. We try to catch them all before they actually stop, and they'd be on and collect what regimens they're on before they stop, at last CD4 count, and then when they passed away. And then we get as much clinical data on them as possible, and I'm going to talk about these particular individuals, but you can see here's LGO1 on some regimen we didn't know about, but then we find out which ones they're on and which one, and then we get their CD4 counts and viral load data. Stephen Rawlings is very good at helping us come up with these very nice figures to figure out what their natural history has been. Same thing for this person, viral loads, multiple agents, etc. So it can be quite complicated in their natural course of HIV infection in modern and pre-modern antiretroviral therapy. So these are the individuals that we're very interested in for this next study, which was to look at the HIV dynamics and repopulation of reservoirs in the human body. And this is work by a lot of us, especially led by Antoine and Sarah Gianella. So I'm going to take their slides today and pass it off as my own. But here we have the sampling, we do the DNA extraction, we do limited dilution, single genome sequencing, we do full-length envelope, we look at those that are intact, we look at those that are not intact, and then characterize, especially the ones of the intact populations, within compartments, and then we look at dynamics using phylogenetics and discrete diffusion models to figure out how the virus is populating all these different tissues. So one of the first questions that we'd have is exactly what is the size of the reservoir? So we measure all these different tissues over here, so cardiac serum, frontal lobe, ileum, jejunum, all across the body, and then start to come up with a map of how many different copies of HIV DNA is there, how many of those are intact, how many of those are not intact. And overall, this is what we found, with the lowest HIV DNA being within the brain. But it's there. Then we can look at the diversity, so we can make a map of the diversity of HIV in individuals. And these are two people who decided to stop their therapy, so I'm going to talk a lot about that in a second. Most of the rebounding populations in the blood, interestingly, were intact, and they were all 99, they were basically identical when they rebounded. But we can also see clonality, these individual identical sequences popping up across tissues, such as in the ileum, 5 out of the 5 sequences that we got that were intact were completely identical. And the same for the testes. And then in the sequencing component, we recovered 605 full-length envelope sequences. Otherwise defective viruses were about 10% of the population. We got about 7 sequences per compartment. No defective sequences were found in the blood plasma of those who were rebounding. And then we did recover, especially from Magali and Carol's work, CNS tissue proviruses in the frontal and occipital lobes. And this is just a tree. When we put all the sequences together, we don't have any contamination, and they all cluster within a person. Sorry. So these are people who stopped antiretroviral therapy, and this is LGO1 from one of our first participants. And these are all the different places that we sampled, blood plasma, blah, blah. There's lots of colors here that people tell me, although I don't see those colors. But if we put those, and y'all who know me, I don't really see colors, but Antoine says that there's lots there. And we can see when we make a phylogenetic tree that there's lots of clonality or identical sequences in different places, and those different places may actually be within different tissues, and that's what this represents. And these are clear for this participant who actually stopped his therapy, that what we saw was a uniform population coming up in the blood plasma, and then we see that exact same population within the tissues that we sample when the person passes away. And then when we look at someone who is suppressed, we see a very similar pattern of, viruses all over the body, but we also see clearly identical populations across several tissues. So there's different colors here, but they all basically have the same virus. So now the question is, where did the virus come from and where is it going? So this is a phylogenetic study using Bayesian methods, and this is Antoine's magic, but you can see all the different places that we might sample, and these are the trafficking, and I'll go a little bit more into that in a second. But here is the Bayesian support for that. On this left-handed side is the source compartment, where we think, using phylogenetic methods, where the virus is going. So like from the blood plasma, I think that's to the liver. PBMCs down here, and we can see these little tracks where things go. So sources here would be PBMC, frontal motor cortex, lymph nodes, blood plasma. So we could just see that there's evidence of migration events during when someone stops their therapy from blood plasma to liver and lymph nodes. Looks like repopulation happens relatively quickly. Here's another person who stopped his therapy. We had 2 of those in this study, LGO1 and LGO4. LGO4 had a much higher viral load once he stopped therapy. So lots of different things going on here. But we actually saw in this particular person evidence of migration within the CNS, specifically from the occipital cortex into the frontal cortex. So the blood very clearly shows that it's the main highway for HIV getting across, but even during rebound that there might be intra-compartment exchange, even in places like the CNS. In a person who remains suppressed, we can do similar methods. We get a smaller snapshot of tissues, but here, here we can see that even within the brain there seems to be lots of exchange in this very small compartment of virus going back and forth. But across the body we get very nice maps. And the patterns remain mostly consistent, that the blood is the major highway for virus to get around, but the gut tissues, and specifically the gut, more than likely are a big reservoir for virus that eventually goes throughout the body. We then wanted to know what were the factors for some of these viral dynamics, and the variables that we looked at were how many viruses that we got from each compartment, what was the level of HIV DNA that was there, what was the diversity. We also looked at X4 tropic provirus in each compartment, and then we looked at various analyses to figure out if this actually was true or not. And this is a generalized linear model, but basically what we saw was that viral dispersal was associated if there was low viral divergence between compartments in about half our people. And then higher viral diversity at the recipient site seemed to be about 1/3 of our participants. And then X4 trophic virus in one of our participants seemed to be consistent with or significant for viral dispersal from that site in one person. So our conclusion was that we had an emergence of large clonal intact viral populations after stopping therapy, which we knew, but those were very identical. They just looked like there was one population that emerged. It was very much intact, and it looked like it repopulated tissues throughout the body. It also looked like there were multiple sites that could act as hubs for the source for the dissemination of viruses within the body, and these mostly occurred within the blood, gut, and lymph nodes. But viral exchanges did occur within brain areas, even within brain areas, and across the blood-brain barrier during when rebound happened, and that the viral dynamics are associated with low divergence between sites and high diversity within an HIV recipient site. So that was really all of Antoine's work to really come up with trying to make out that map of where all this virus goes. One of the things that we really are struck by is that the blood is the highway for the virus to get across, and it might explain some of the things that we've seen with the Timothy Brown, the Berlin patient, and the new London patient, where their blood has basically been protected from getting HIV, so that perhaps there might be reactivation in all these sites. And Doug was one of the first ones to point out this. We didn't do anything to reservoirs and deep tissues, we just did it in the bone marrow. How is that keeping virus from taking hold, right? Well, maybe it is the highway that is important. If you protect the highway, then you might still have reservoirs in all these sites, but it doesn't get to all these other places. One other one, just as a tease, we're very interested in now, since we have tissues that are so fresh, we can actually start imaging them. So this is work with Colin Kiefer, who will be here soon. As a plug for CFAR, is going to bring him down to talk about his imaging techniques. But basically, we can take tissue, colon, spleen, duodenum, ileum, and he has ways to sort of get it to the point, take out collagen, so that we can image it. Here to orient you, these are the villi within these different gut tissues, and when he stains, he can stain with DAPI and CD3 and macrophage, and to look at all the different cells that are there. Well, he gets pretty good staining, but then you can look at immunofluorescence as well. And basically, with all the colors again, gosh darn it, you can see all the live cells, but in fact, you can actually go down deeper to figure out where the T cells are, where the macrophages are, and even where the P24 are. And this is someone who has actually remained virally suppressed, but you can start to localize where the P24 is in close to CD4 T cells, and then to actually look at it in a 3D structure of what's actually going on within gut tissue or brain tissue and hopefully other tissues to get a better sense of that. So before we move on, this is the last gift for these individuals, and this is a big important thing for these people. They're finding great meaning at this time at the end of their life to participate in HIV research. But some of those questions that I asked early on were questions that we're asking these people as well. I had a call with the FDA a couple of days ago with Lior Weinberger. I don't know if y'all remember him, but he was here at UCSD. And he's developing these new transferring infectious particles that he wants to test. And he's done some in mice, and he's done some in monkeys, but he really wants to start testing them in people. And that goes back to that last question that I asked. Would you be interested in being exposed to an infectious agent at the end of your life for science, even though you might be infected with those? So he proposed that perhaps this infectious particle, we could start testing it in humans, and he's trying to work on a DoD grant, but the FDA had to get involved. So we had this 1 hour and 30-minute call scheduled for them, and within 15 minutes, they're like, oh no, we think that's fine. You should go ahead and start working on that. And what that means in reality is, if this is true, you could really change how we start to do biomedical research in people. Understanding who's really vulnerable and who's really not vulnerable to be able to consent for things on their own, but skipping out a lot of monkey studies that may not have the same cellular pathways that we do, or respond to various infectious agents the same way that we do, might really be able to spur some biomedical development. So that's one last gift I think that this cohort promises to do. So acknowledgements, very much thank all of our participants. Our community advisors have been some of our biggest advocates. The whole reason that the FDA heard us out in the first place is that the community advocates went to them and said, hey, this is something we're very much interested in participating in and want to push it. And that's how we got the way into the door the first time. And then of course, this is the last gift team, a lot of people in this room, and Antoine being the real and Sarah being the real driving forces for the data that I presented about the dynamics. Okay. That is the end of our ceremonies. So thanks.